have nuclei. So bacteria and archaea, they lack nuclei. That's a basic definition of a prokaryote. Here are some examples. Don't try to memorize the shapes and structures. We're going to be talking about that in the laboratory this week, and then next week, actually these tiny microorganisms to visualize into the microscope. All right? Just a little bit about archaea. We don't study them too much in this class. They're very fascinating. Let me just say they don't cause disease in humans. The major reason why they haven't adapted to our system. Let me give you a sense of what these organisms are. Again, they're single-cellular organisms. They lack nuclei. They are prokaryotes. If ever you've been to Yellowstone, you probably saw those hot vent areas like those pools and bubbling and stuff. And then you look on the base of the pool, you see different colors, those gnats. Those are the bacteria. Those are archaea. So archaea, these are actually I shouldn't say bacteria. Archaea are single-cellular prokaryotes that live in some very strange environments. High temperature, low temperatures, high salt, low salt, acids, bases, and they're very resilient. They're ancient. Let me just say, some of them, they can't even grow if the temperature is not 80 degrees centigrade, not Fahrenheit. All right? Some of them can't even grow unless water's boiling. Think about that. So they've adapted to the environment over millions of years, probably, but they don't cause disease in humans. The ones that do actually belong to the bacterial group. Viruses, of course, they're not living organisms. They are what I call molecular parasites, cause many different diseases. They are all what we call obligate intracellular parasites. And again, don't worry about terminologies. We're going to get to that when we go to each of these groups. All right? I'm just trying to get you a sense. So they require a living cell to grow. There's a cell. This is the nucleus, actually, and these are viruses replicating inside the cell. You can't grow viruses just like you grow E. coli. E. coli, you can just start a culture growth. Overnight, come back as billions of cells. Not so for viruses. Viruses require living cells to grow. All right? That's a smallpox virus, by the way. Fungi, we've already seen, we probably come in contact with them already. Think about bread mold, that green stuff growing on fruit. Those are fungi. Let me just say, many fungi are very useful to us. They produce antibiotics. And then I'll talk about one in particular, penicillin, later on. But very few fungi actually cause disease in humans. Really, most fungal diseases in humans only involve about 12 genera of fungi, whereas for bacteria, have hundreds of them. All right? Protista, so everything other than bacteria and archaea, they are eukaryotes. Eukaryotes have nuclei in their cell. So protista, again, don't worry about memorizing names at this point. This is a blood smear that was taken from an individual with a disease called African sleeping sickness. It's not caused by a bacterium, it's caused by a parasite. So what you see on this slide, right over, that's a red blood cell. These other ones here are white blood cells. And these little squiggly things, that's a single cellular organism, all right? It's called trypanosoma. There's something called American trypanosome, very different disease. We're going to look at that later on again when we talk about parasites. But this is the organism that causes African sleeping sickness, a very strange disease. The disease is transmitted to an insect called the tsetse fly. Bites you, injects the parasite into your bloodstream, and then this one over a month, depending on the species, a few weeks to a few months, penetrates to the blood-brain barrier. It gets into the brain, destroys brain tissue, and pretty soon, it takes about 3 to 5 years, generally speaking. They slip into coma. There comes a point when you can't return that person to health. In other words, that person will die. All right? So that's African sleeping sickness caused by trypanosome brucei. Don't worry about names yet. Trying to get you a sense of what these things look like. All right, so around this time, other things are happening. And again, I'm not going to talk about DNA structure and all that stuff, but we're learning a whole lot in a very short period of time. But you see how the history is developing. Well within 100 years, in fact, well within about 75 years, we learned so much, even what is our genetic material, which turned out to be DNA, by the way. That was only discovered and proven once and for all in 1952. And that's not a long time ago, all right? In fact, I was about minus 10 years old. I was born in 1962. That stuff was discovered in 1952, the structure of DNA. All right, so let me summarize this for you. Eukaryotes are organisms that have nuclei in their cells. Your cells have nuclei. Parasitic cells, parasites, worms, for example, they have nuclei. Fungi have nuclei. Proteins have nuclei. Those are eukaryotes. So plants, animals, protists, and fungi, they are eukaryotes. Basic definition, this is basic as we get here. There's a nucleus in those cells. Prokaryotes, remember these are bacteria and archaea. They're really ancient organisms, especially the archaea. They go back way back in history, a couple billion years ago. They lack nuclei. Okay? All right, so moving on to some other incredible individuals. Paul Ehrlich, he's known as the father of antibiotics. Really, check out what he did. And again, his work goes back to 1910. By the way, I told you these guys' lives overlap. Let me just say, he worked with Robert Koch, by the way. He worked with Pasteur. They all knew each other. Well, he didn't work with Pasteur. He worked with Robert Koch. That's the major one. All right? So, in fact, he was his student per se, mentor. But here's what he's interested in. He wasn't interested in microorganisms per se, but it turned out his work actually gave him that interest in microorganisms. He was interested in blood cells. How do we classify them? How do we characterize them? Again, this is over 100 years ago. So you picture his lab. There's lots of stains all over the place. And here's what he noticed. When he uses a stain and he stains cells, your cells, white blood cells, the cells die. So he's thinking, man, chemicals can kill life per se. Of course, we know that to be true now. Well, around this time, there was an epidemic of syphilis. All right, so this is turn of the last century. Tons of people with syphilis. And he said, you know what? Can these chemicals, they kill white blood cells, but can they also kill bacteria? So imagine here's his lab. He has all these compounds all labeled. In fact, he comes across compound 606 as it's called. All right? Many different chemicals in there, but it contained asphenamine. This is an antimicrobial. So he got patients to volunteer, they come into his lab, and he injected them with this dye that contained asphenamine. And guess what? That dye, out of the thousands that he had, killed the organism that caused syphilis. Now, the organism that causes syphilis is Treponema pallidum. And remember, we can't grow that in the laboratory. So again, Paul Ehrlich, the first one to show that chemicals can kill microorganisms. Not only microorganisms, but microorganisms that cause disease in humans. All right? Very important observation back over 100 years ago. And by the way, after he published his stuff, man, governments just swarmed him. They wanted to get that chemical from him, from his lab, so they can treat people with syphilis. But he was very careful. He did give it to them. But here's the reason why a few people died. And this happens for whatever reason, maybe reaction to other chemicals in that preparation that he had. But there's always collateral damage. Antibiotics are a good thing. But some antibiotics, if it's taken too long, can actually cause kidney problems, liver problems. All right? Nevertheless, his work actually made pharmaceutical companies just make a big search in the environment for organisms and for chemicals that we can use to treat diseases, microbial diseases. Now, the last guy on the list is very important also. His name is Alexander Fleming, and his notoriety goes back to 1929. All right, so try to picture this. He's working in his lab, and here's this plate. This plate is growing bacteria. That stuff in white, that's Staphylococcus aureus. And just by chance, he was careless one day. He left the lid open, and a spore of this mold landed on the plate, and it grew. You see it smack in the middle right over here. That mold is a fungus. It's Penicillium notatum. So this is a fungus that, here's what it does. It produces penicillin. Penicillin diffuses in the agar, whereas everything was white here indicating bacterial growth. If you notice, there's this huge zone. It literally obliterated a bacterial cell. So penicillin literally killed the bacterial cell, so you see this clear zone. That's the first time it was shown that a living form, in this case penicillium, a mold, can, right over here, that's the mold, Penicillium notatum, can actually produce something, in this case it was penicillin, that would kill bacterial cells. Let me just make a little comment on penicillin. Fleming was not a biochemist, so he could not isolate the drug, penicillin. So he teamed up with two biochemists, Flory and Cheng, and they were able to purify penicillin. Let me just say this. This is 1929, right? World War II began in 1939. Within 10 years, they had that drug. Without penicillin, there have been millions of more deaths because of World War II. And of course, you use it for other diseases. Imagine somebody's on the battlefield, they're being stabbed, you have soil and wounds, lots of infection and disease all over the place. Without penicillin, you'd have millions of people who would be dead during that wartime. All right? Okay. So, what are we dealing with today? All right. A good example would be COVID-19. It's not a microorganism. It's not a microorganism because it's not an organism. Viruses are molecular parasites. They're not alive. All right? But still, we can use that as an example to show you what I have illustrated on the slide. So the thing that we're dealing today that really caused disease to spread, of course, right over here, global, several different things, but global travel. Think about COVID-19. If you know the history of that, the first time we saw this, humanity saw that was in Wuhan, China. This goes back to last December, or maybe a little earlier too, right? And then because people were traveling all over the place, you take an infectious agent from a local area, you have global travel, and now you're spreading that infection all over the place. Turns out that we have a quarter of all the cases of COVID in the United States, right? Began in Wuhan, China. And of course, this spread worldwide. It's one of the major things we haven't seen for probably centuries. Do you realize what happened? Let's just go back a few months. You have to shut down everything. I mean, here's an infectious agent, literally caused humanity to almost come to a standstill. One agent, terrible disease. Of course, we're going to look at something that's related to it, but not as vicious. It's called SARS, that occurred back in 2002, again in China. All right? So global travel allows disease to be spread. But nobody's going to ban global travel, for example, on a long-term basis, because we're humans and we travel to experience culture and commerce and all that stuff. But nonetheless, global travel is a major route, a major mechanism to transmit disease around the planet. Now, there are things that we call emerging diseases. Now, these have been around for a while, but they're not ancient. For example, HIV and AIDS. That came on the scene big time in the early 80s. Well, you might say that's about 40 years ago. Well, in real time, right? Let's say geological time, that's just a drop in the bucket. That's like a grain of sand as far as time goes. So those are emerging diseases. COVID-19 is an emerging disease. SARS is an emerging disease. MERS is an emerging disease. HIV, AIDS is an emerging disease. Ebola, first time we saw Ebola was in 1976. That's an emerging disease. And then we have diseases that we call re-emerging diseases. There's a long history. Think about tuberculosis. It's been around forever, right? Leprosy in some areas of the world, all that's getting a lot better today. But they're ancient diseases, or very old diseases that we see and re-emerge. Even syphilis is on the list, by the way. Syphilis has been around forever. But we see an incline just right after 1980. Basically, a lot of AIDS patients, immune system compromised. Other reasons why, promiscuity included, by the way. If you go back to 2 summers ago, big epidemic. Guess where? In Palm Springs, California. The biggest epidemic that year of syphilis occurred in Palm Springs. So those are re-emerging diseases. They've been around for a long time, and now they subsided for a while, and now we see an increase in the number of cases. All right? Our current challenges, let me just list a few for you, many of them actually, including some I've already mentioned. Zoonotic diseases. So what is a zoonotic disease? Well, you go to the zoo to see animals. There are many diseases that are transmitted to humans via animals, insects included. Ebola in bats, pangolins and bats and COVID-19, the SARS virus, Zika virus and mosquitoes. And again, don't worry so much about memorizing these things. We're going to get to it later on. But I want to broaden your perspective on how diseases are transmitted and how we are challenged by those transmissions. All right, West Nile, right in the United States, transmitted by mosquitoes. Malaria transmitted by mosquitoes. Dengue, yellow fever transmitted by mosquitoes. The Black Plague caused by Yersinia pestis transmitted to rodents and fleas. The rodents, like rats, they have the pathogen in them. Fleas bite them, and then the fleas are infected and they bite you. No, you have disease. Another thing that we're challenged by is increasing resistance to antibiotics. Now, this is pretty big. And I'll give you one example. We probably have all heard about MRSA. MRSA. That stands for methicillin-resistant Staph aureus. All right, many different strains of Staphylococcus aureus. But MRSA, many different strains of MRSA also, but that's a very resistant organism. So it's many times the last antibiotic used to treat diseases caused by that organism, it is something called vancomycin.